Plasmax is a chemically defined, protein-free culture medium based on the nutrient profile of human plasma. Unlike traditional media, Plasmax contains growth-stimulating trace elements that promote growth in mammalian cells. In this video clip from Cell Bio Virtual 2020, Plasmax developer Dr. Saverio Tardito explains how trace elements in Plasmax produce colony growth in triple-negative breast cancer cells that outperforms DMEM at low serum concentrations.
So what do you do the first time you try a medium? You want to know if your cells are growing or not. And that's what we did. We compared the cell number of cells incubated in the two media after 3 days of incubation. And there was not significant difference between the two media in all three cell lines. And this is a good news, because it means that even if we are reducing the concentration of all the nutrients we supply them, the growth is supported and sustained by the medium in a comparable way with historic medium. But this was not true when we placed the cells at much lower density to perform a colony forming assay. As you can see from this image, all three cell lines, and in particular the MDA-MB468 cell line, grew better in Plasmax than in DMEM-F12 when a colony forming assay was performed. And this is the three experiments are quantified here with its statistics. Okay, so we were particularly interested in this phenotype, and we wanted to understand which of the component of Plasmax, or perhaps which of the component of DMEM-F12 was favoring or inhibiting, respectively, the colony forming capacity of these cells. And so we took a methodic and systematic approach. And first we mixed the two media 1-to-1, and we observed if the colonies were able to form or not. And the answer was yes. And this put us on the right track, that something present in Plasmax was favoring the formation of colonies, rather than something missing. Sorry, that something present in DMEM-F12 was inhibiting the capacity of forming colonies. And then we dissected the composition of Plasmax into various components, stock solutions, and to our disappointment, because we are a cancer metabolism lab, and we would have much preferred to find an organic component to make the difference, we found that it was an inorganic salt. So we look at the differences between the salts present in DMEM-F12 and Plasmax, and we identified sodium selenide as the compound, the salt responsible for the capacity of the 468 cells to form colonies or not. And this was clearly demonstrated by this dose response curve shown here, where about 5 nanomolar of selenium were sufficient to stimulate the colony formation of these cells.
As you have heard from Dr. Tardito, Plasmax is the only ready-to-use medium containing growth-enhancing trace elements at physiological levels. You can access Plasmax through the Zenbio website.